And our final speaker closing out this awesome day of science is Hayley. And Hayley's going to talk to us about genetic diversity in BC reforestation, a closer look at effective population size. Where are my slides? Oh, they're over here. Yeah. And then you have to like. You got it. Oh, got it. Okay. Well, thanks everyone for sticking out until the end. I already really like this conference, but everyone being here makes this my favorite conference ever. I want to start off by acknowledging that UBC sits on the traditional and unceded territory of the Musqueam people in what we now call Canada. And I'm going to talk to you, like has already been said, about effective population size in BC reforestation. So here in BC, the generations are starting to increase in the selective breeding programs at the same time that the percentage of landscape that is harvested and replanted with the selective seed is increasing. And it's important to remember that these landscapes are a mosaic of planted and natural stands. And so this has led both managers and the public alike to ask what the consequences of this forest management is for landscape level genetic diversity, especially because diversity at this scale will be incredibly important for resiliency to climate change for these forests. And so the first step in addressing this question is how well is genetic diversity maintained within the breeding program? And here in BC, it is monitored and maintained at the seed lot level by estimating effective population size from parent tree contributions in the seed orchards. And so we want to know, how do these estimates compare to genomic estimates? So how are these boots on the ground estimates comparing to genomic estimates? As well as how genetic diversity in these selectively bred seed lots compares to genetic diversity in natural seed. And so we address these questions by looking at two BC forestry species, Latchel pine and interior spruce. And I was lucky because there's already some great genomic data sitting there, which is your dream as a postdoc. And so we looked across panels of roughly 1,200 putatively neutral and genome-wide SNPs, comparing both selectively bred lots as well as natural lots, so seed that's taken out of these natural stands. And we compared genetic diversity estimates in natural stands versus selectively bred lots, as well as comparing different estimates of effective population size. So we have that boots on the ground estimate using gamete production, and we compared that to two estimates from genomic data, one using linkage disequilibrium and one using SIB-chip frequency. So looking at the number or frequency of siblings from pedigree reconstruction. And the results were quite similar across interior spruce and lodgepole pine. So I'll just be showing you the results from lodgepole pine. And so when we look at these typical estimates of genetic diversity, expected and observed heterozygosity and inbreeding coefficients, and we're looking across natural in green and selectively bred in blue, we see that the estimates are quite similar and that they are not significantly different. But these differences start to really emerge when we look at effective population size. And so on the left, you will see our selectively bred lots. And I'm not showing you the natural lots because most of the estimates were either at infinity or approaching infinity. So this is where we really start to see differences. And then when we look at that second bit where we're comparing the different estimates of effective population size, we see that our gamete production estimates in orange are quite similar to our two genomic estimates in green and blue. And most times they're actually underestimated, which is really great news for genetic diversity. It means that what's going on in the landscape is more than what had been the minimum for monitoring. And when we look where that gamete production estimate is less similar, and we look into the characteristics of our seed orchards, we can start to understand why. If it's underestimating, it could be down to pollen contamination based on where that seed orchard's located. And if it's overestimating, it could be down to forward selection and the equations that are used to calculate that effective population size. So now we have a pretty good understanding of how genetic diversity is maintained within these breeding programs, but that does not fully address the question of what the consequences of forest management are for genetic diversity at this broader scale. And so the next step is to look at the shift in genetic diversity from orchards to seed lots to actual planted stands on the landscape. And then we'll be using the results from step 1 and 2 in a landscape genetic simulation model, modeling these natural and planted stands that are interconnected through gene flow on the landscape and varying parameters that are associated with this forest management in order to look into the future and really understand the effect of forest management on genetic diversity at this broad landscape scale. So keeping it short and sweet. If you have any questions, let's chat. Come find me or email me or tweet at me. But if you have any questions, comments, or just really like conservation genetics, I'm down to talk. And I would really like to acknowledge all of my great collaborators. Sally Aiken is here, and this is an incredibly collaborative project that is tightly linked with both industry and the Ministry of Forests in BC. Do you have any questions for me?
No, I'm failing as moderator, not thinking of it.
That's fine. It's quite late.
We spoke about it a lot.
No, we didn't.
Could everyone join? Oh, sorry.
So, what's your recommendations to the tree breeders in those two species given the results that you see so far?
Yes, that's a really good question. I was expecting someone to ask, and we're still in that discussion phase because it's not obviously Sally and I giving recommendations to the tree breeders. It's us working with the ministry and getting input from the breeders in order to come up with what the next step is. And so, yes, we now know that unsurprisingly, the effective population size is lower in these selectively bred seed lots. But what does that really mean? And so I think that the simulation modeling is going to really help us better understand what the true consequences for that are. I mean, BC already has a minimum NE, which is great, ahead of the game in a lot of ways. And so whether or not that minimum NE is like a good minimum is really going to come at the end of this project.